Hello everyone. In this lecture, you will learn about bacterial transformation. Every part of this lecture is extremely important, so make sure you don't miss any point. So, what is transformation? It is the process by which a bacterial cell can take up genetic material from its outside environment. So if this is a genetic material in the extracellular medium of the bacteria, it can take up that genetic material within its own cell by the process of transformation. Transformation. So obviously the question arises that where does this extracellular genetic material come from? In the natural transformation procedures, and I should emphasize the term natural because we'll encounter another type of transformation, that is artificial transformation, where we perform the transformation in a laboratory setup where we supply the genetic material. But in the natural transformation, in the natural environment, these genetic material, they come from dead bacteria cells. So for example, if this is a petri dish and this contains a colony of bacteria, so there will be lots and lots of bacteria in this colony. Now due to some reason, if a bacteria dies, it releases all its intracellular content in this extracellular media, including the genetic material, that is its DNA and plasmid, etc. So those bacteria that are still alive surrounding this dead bacteria, they can take up those genetic material that were released from that dead bacteria using this process of transformation. So one term you will encounter a lot in transformation is horizontal gene transfer. So what is horizontal gene transfer? To understand this horizontal gene transfer, we should first understand what is vertical gene transfer. So let's study what is vertical gene transfer. What happens during a cell division in a bacteria, that is the asexual reproduction of a bacteria, is that a mother cell containing its genetic material, it divides into two daughter cells. So if these two are the daughter cells from that mother cell, they will contain exact copies. If there is no mutation, then exact copies of that maternal DNA within their own cell. So these two are daughter cells. So in a way of saying, the genetic material of the maternal cell were transferred vertically within the daughter cell. This is known as vertical gene transfer. So what is horizontal gene transfer? In this method, two bacteria within the same generation can transfer genetic material or can exchange genetic material with one another. So as you may have noticed, in vertical gene transfer, the mother and daughter are of course in two different generations. But in horizontal gene transfer, the process is within two bacteria of same generation. There are various types of this horizontal gene transfer, and transformation is one of them. So the types of horizontal gene transfer. The first type that we are discussing now is, of course, transformation, in which the bacteria can take up extracellular DNA within its cell. The second process is called conjugation, in which two bacteria, one is called donor bacteria and another is called recipient, they can transfer genetic material from the donor bacteria to the recipient bacteria using a tube called conjugation tube or mating bridge. So the genetic material gets transferred from donor cell to the recipient cell. And the third process in the horizontal gene transfer is called transduction, in which the bacterial cell receives DNA from a bacteriophage or virus that can infect bacteria. So the genetic material gets transferred from the bacteriophage to the bacteria. Another question you might ask is if there is any similar process of bacterial transformation in the eukaryotic cell. The answer would be yes. There is a similar process in which an eukaryotic cell can take up genetic material from outside. But this process in eukaryotic cell isn't called transformation. It is called transfection. So why the different naming? This is because in eukaryotic cell, the term transformation is used to describe a specific stage of carcinogenesis that is called malignant transformation. So to avoid confusion, this similar process of bacterial transformation was named transfection in eukaryotic cells. One thing you should always remember is that the bacterial cells that undergoes transformation cannot do this all the time or any time. They can undergo transformation procedure only when they are competent. And the process by which a bacterial cell become competent or able to undergo transformation procedure is called competence. A bacterial cell is not always competent. So a bacteria cannot undergo transformation anytime it wants. And also once it becomes competent, it doesn't stay competent for very long. So the state of competence is transient, during which the bacteria can undergo transformation. It is kind of like window of opportunity for undergoing transformation for those bacteria. So when does a bacteria becomes competent, or what factors decide when a bacteria will undergo competence? So these factors vary widely among the bacterial species. Some bacteria, for example, are naturally competent. So they can become competent and incompetent several times in their lifetime and without any specific trigger. So these bacteria are naturally competent. One such example of this bacteria is Neisseria. Various Neisseria species can become competent at various stages of their life cycle. Another factor that determines competence is stress, and it is a widely accepted factor in determining competence. So, to understand how stress can induce competence in bacteria, you should first ask why bacteria undergo competence in the first place. What is the evolutionary significance of a bacteria undergoing competence or doing transformation? So, this is due to the fact that during transformation, the bacteria can gain new properties that might help it with its survival from the genetic material that it takes up. So, if a culture media contains various bacteria and some of the bacteria contains an antibiotic resistant gene in its DNA, particularly the plasmid DNA, and if the bacteria dies due to some reason, it will release the DNA into the extracellular media, and this DNA will contain the gene for the antibiotic resistant. Now the other bacteria that are not antibiotic resistant, that is they are antibiotic sensitive, so these bacteria can take up the antibiotic resistant gene within their own cell and incorporate that gene in their genetic material. This way, the bacteria can become antibiotic resistant. So, the transformation is helping this previously antibiotic sensitive bacteria in its survival when it gets exposed later to that particular antibiotic. So, transformation in this case helping the bacteria in their survival. Now come to the factors. During stress, the bacteria is in a situation where it may undergo death. So the bacteria is in need of something that will help it with its survival, and transformation is a procedure that might help them in their survival. So stress is a very well-known factor that triggers bacteria to undergo transformation by making them competent. So what are some of the examples of stress? When we take a petri dish containing strep pneumoniae, bacterial culture, and we apply a sublethal dose of aminoglycoside or fluoroquinolone antibiotic within this culture media, then we find that many strep pneumoniae bacteria undergoes competence. So this sublethal dose of aminoglycoside or fluoroquinolone, they does not kill most bacteria, but they help them undergo transformation. Another example is UV radiation. So as we know that UV radiation can cause pyrimidine dimer formation. So it can trigger mutation or at least DNA damage in parts of the DNA containing pyrimidine. So this is one form of stress, and UV radiation can trigger transformation or competence in again strep pneumoniae and Legionella pneumophila. Another factor that directly causes mutation or damage in the DNA is mitomycin C. So this is one of those chemicals that can trigger formation of cross links in the DNA of two DNA strands. So this mitomycin C also triggers competence in Streptococcus pneumoniae. Another form of stress is exhaustion of nutrient in the surrounding media, which triggers competence in Haemophilus influenzae. But this trigger does not work similarly in all species of bacteria. For example, presence of high level of nutrients can trigger transformation or competence in various species of Acinetobacter. So we categorize nutrient as a separate factor. There is also presence of various specific nutrients that can trigger competence in certain bacteria. For example, presence of casein protein triggers transformation or competence in Streptococcus thermophilus. Another very important factor that determines competence is cell population density or quorum sensing. The bacteria contains certain genes that can sense the population density in the culture media or in the surrounding, and they trigger the activation or inactivation of certain genes depending on the certain population density in its environment. For example, in Streptococcus pneumoniae, the bacteria becomes competent when there is an exponential growth phase. Particularly the early exponential growth phase. Similarly, in Bacillus subtilis, the bacteria becomes competent when it undergoes stationary phase. One thing you might have noticed that most of the examples contain either strep pneumoniae or Bacillus subtilis. This is because most of the research on transformation were done in these two bacteria species. Also, nowadays competent bacteria are available commercially, so we can buy those commercially available bacterial strains that will very easily undergo transformation and perform various experiments on transformation in the laboratory setup using those bacteria. So once the bacteria is competent, it can undergo transformation. But how it undergoes transformation? So there are actually two general classification. One is natural method and one is lab method or artificial method. So in the next video we shall discuss the various natural methods of transformation, and in the video that followed we shall discuss the artificial method of transformation. So if you found this video helpful, please like this video and also subscribe so that you don't miss any future update. If you have any question regarding this topic or any other topic related to biology, you can ask them in the comment section and we shall try to answer as many of them as possible. Also, if you have any suggestions, put it in the comment section down below. For the links for further reading or references like research paper in this particular topic, visit the description section of the video. So see you in the next video. Thank you for watching.